Hi everyone, welcome to week 1 of general biology. I'm so excited to have you guys here. We're going to start off with a little introduction to general biology, talk about some of the broader concepts that we might be covering more in detail throughout the course of the semester, and go from there. So just starting off a little bit about me, my name is Dr. Beck. You can refer to me as that. I first got my bachelor's in biology with a concentration in pre-medicine at Edinburgh in Pennsylvania in 2015. I then went on to get my master's in applied clinical nutrition and my doctor of chiropractic at New York Chiropractic College in Seneca Falls, and I graduated early last year. So now I'm working as a professor, and I also am working as a chiropractor, starting up my own practice. I work at a couple of different schools teaching. I teach here at FLCC, I teach at St. John Fisher, and I teach over at Bryan and Stratton. Currently, I live in downtown Rochester. I live with my 2 pets. I have a little mixed terrier breed, Remy, and I have a tortie cat. Her name is Sydney. In my spare time, I like to read. I read a lot of books. I am learning Spanish and sign language. I do some powerlifting. I also do some painting. So when we start looking at biology, we have to first look at the natural sciences. So if you ever go to a natural history museum or science museum, generally it will encompass all of these different branches of science. Natural sciences can be broken down into life sciences and physical sciences. Life sciences being the studies of living things. That could include things like biology, but also things like zoology, entomology, ornithology, anything that has to do with living things. Physical sciences are the non-living things. So those are things like astronomy, geology, physics, chemistry, and altogether those encompass natural sciences. Biology is very applicable to day-to-day activity. You hear about biology all the time in the news and media and social media. Currently, you hear a ton about COVID-19 that has to do with biology. You hear about E. coli outbreaks, climate change, microbiomes, what probiotics you should be taking, what foods you should be consuming in order to have a healthy digestive system, healthy lifestyle. These are all things that you might be exposed to on a day-to-day basis, and they're all considered part of biology, because biology is the study of living things. It's the study of life. So as we're going through this semester, you might notice that we cover a really broad amount of topics because the study of life is a very big, overarching subject. The microbiome is considered the bacteria that live either on you or in you that help to maintain your daily life. I posted an article for this week that talks about the microbiome in great depth and how it can assist in some disease processes to make you healthier. You have to have a really healthy microbiome to have a healthy digestive system. You have between 10 and 100 trillion symbiotic bacteria that either live on you or in you at any given point in time. So what it means to be symbiotic is that these bacteria are here to help you. These are good bacteria. You want these living either on your skin to fight off bad bacteria that your body comes in contact with, or within your digestive tract to fight off bad bacteria you might consume. So make sure you read that article this week. It's really interesting. One of the main concepts for this week is the scientific method. I'm sure you guys have heard about this. I'm sure you've done a science fair at some point in your life where you had to do the scientific method. You had to study it, and you had to do an experiment based off of a hypothesis. So the scientific method itself was first documented by this gentleman here on the side, Sir Francis Bacon. He believes that knowledge and learning are derived from inductive reasoning. So we'll talk about the difference between inductive and deductive reasoning later on in the lecture. His thoughts about inductive reasoning are a bit incomplete because we need deductive reasoning in order to make conclusions about cause and effect when it comes to experiments and what they might mean. The first thing that happens during the scientific method is observation. You might observe a phenomenon going on and make a hypothesis based upon that. An observation could be that you put a piece of toast in the toaster, but it didn't seem to get toasted. My toast is not toasted could be your observation. Your hypothesis could be, my toaster is not toasting my bread. So in order to test the hypothesis, you could then run through some experiments where you test different settings on your toaster to see if there are different settings that work and different settings that don't. And you could use that with a control with a different toaster. So what makes a good hypothesis? A good hypothesis should be testable. So you should be able to test your hypothesis. If it's not testable, it can't be a hypothesis. You have to be able to run experiments to see if your hypothesis is supported or non-supported. So if your hypothesis were that there are ghosts in your house, that's not exactly a testable hypothesis. Some may say that that's true, but in the sense of the science community, it's really hard to make experiments based off of supernatural things. Your hypothesis should also be falsifiable. So saying simply a fact is not a good hypothesis. For example, the sky is blue. That can't be either testable or falsifiable. It should also be known that correlation is not the same as causation. This is something you might hear a lot in the science community. So when we look at the experiments and if the hypothesis is supported and it's just because there's a conclusion where two things are correlating does not mean that it's good to generalize a conclusion about it. For example, two variables that are often correlated that don't seem to make a lot of sense to people is that the higher the ice cream sales, the higher the crime rate. And you might be thinking that there's no way that those two variables could be related. That's because they're correlated, whereas one goes up, the other goes up, but that doesn't mean that one causes the other. The increase in ice cream sales does not cause the increase in crime rates. In fact, the reason that they're correlated is because of the time of the year that ice cream sales go up, and that's in the summer. Summertime means higher crime rates in general, and that's just a statistic. Another distinction that should be made is between a hypothesis and a theory. A lot of people use these terms interchangeably, and they shouldn't be used interchangeably. A hypothesis is a proposed explanation for a phenomenon based on limited evidence. So we're doing an experiment on something that we're not really sure if it's going to be supported or non-supported. It's not scientifically tested or proven yet. There aren't any experiments on it. There's limited data, but it can lead to a theory, because a theory is a set of ideas intended to explain facts or events. It is scientifically tested and proven, and it's based on a wide range of data. It can be formulated through a hypothesis. So the wording here is a little misleading. Just be careful, because theories are not necessarily proven, but they haven't been disproven. So we can't for sure say that they are a fact. They just haven't been proven wrong yet. For example, the theory of gravity. It's called the theory of gravity. We know that gravity exists, but only because it hasn't been proven that it doesn't exist yet. I know that's kind of a convoluted way of thinking about it, but that's what a theory is. It's not a fact, it just hasn't been proven wrong. So a hypothesis can lead to a theory, which is slightly more set in stone than a hypothesis. So we talked a lot about hypotheses, what makes a good hypothesis, how to make a hypothesis, what differentiates a hypothesis from a theory. Now, how do we test a hypothesis? So you have to do an experiment. In an experiment, you need one or more variables. So we need an independent variable and a dependent variable. The independent variable is going to be the variable that's changed in the experiment. The dependent variable is the one that's going to be affected by the change of the independent variable. So if we go back to our example about the toaster not toasting the bread properly, the independent variable would be the toaster settings. That's the thing that we're going to change throughout the course of the experiment. We'll change the toaster settings, and the dependent variable will be the bread, how much it gets toasted. So that will depend upon the changes of the toaster settings. A control group is going to be something that does not change during the experiment. It could be regular bread, and we could compare the bread that we get toasted in the toaster to a regular piece of bread to compare the toastiness. In some experiments, there are multiple independent or multiple dependent variables. Sometimes there's multiple control groups. So it really depends on the experiment itself and what they're testing for. So let's try and look at these examples and try to identify the independent variables and the dependent variables. So these are a list of hypotheses, and let's go through them. So the first one, if I study for 10 hours before my test, I will get an A. So I'll give you a minute. The independent variable would be the amount of time that you're studying. So the 10 hours in this example. That's the thing that's being changed. The dependent variable then would be your test grade. Your test grade depends upon the amount of time you study. Is this a good hypothesis? I would say so. It's testable. You can study for 10 hours, and you can see what test grade you get. And it's falsifiable. You could study for 10 hours, get a test grade that's not an A. The next example is, Wegmans brand paper towels are more absorbent than Target brand paper towels. What would be the independent variable, and what would be the dependent variable? So if you guessed that the independent variable is the brand of paper towel, you would be correct. That is the thing that's changing. Is it Wegmans brand or is it Target brand? The dependent variable would be how absorbent it is. That will depend upon the brand of paper towel. Lastly, it's kind of a weird sounding example, but it comes from a real experiment. So let's see. If a toddler is exposed to a loud noise when he is exposed to white furry objects, he will learn to fear them. If you said the independent variable is the loud noise, that would be correct. The exposure to the loud noise or the non-exposure to the loud noise, that is the thing that's changing. The dependent variable would be the fear of the white furry objects that will depend upon the loud noise. This experiment was called the Little Albert experiment, and it was performed in 1920. It was a psychology experiment based upon irrational fear. Of course, a toddler or any small animals, if they're exposed to loud noises, they'll learn to fear whatever they're being exposed to. So this came with the psychological theory that irrational fears appear in early life. Okay, just to recap about the scientific method. First, you make an observation. Then you ask a question, and usually that question is formulated into a hypothesis. You can make a prediction based on that hypothesis. Is the hypothesis going to be true or untrue? Is it going to be supported or non-supported? Then you do an experiment to test the prediction, and you analyze the results. Is it supported or is it not supported? You report the results, and if it's not supported, try again. So let's talk a little bit about scientific reasoning. Usually it's broken down into inductive and deductive reasoning. Inductive reasoning is taking a general statement and making it more specific. For example, Harold is a grandfather. He is bald. Therefore, all grandfathers are bald. So you can see that there is a little bit of lapse of logic there. You can't always take something general and make it specific. Sometimes it works, but inductive reasoning usually leads to flawed logic. Deductive reasoning, on the other hand, takes something specific and turns it into a general statement. For example, all men are mortal. Harold is a man. Therefore, Harold is mortal. So, in that case, that's a true statement. Unless Harold is a vampire or something. But typically, deductive reasoning leads to a true statement. There are also different types of science that base the way that their logic runs off of these different types of reasonings. Descriptive or discovery science is based upon inductive reasoning. Usually it's more of an observational type science. Hypothesis-based science, or science that runs on experiments, leads with deductive reasoning. So usually the conclusions that come from hypothesis-based science can lead to actual generalizable knowledge. So let's practice. There are 4 examples here, and we just want to know if the example is showing inductive or deductive reasoning. So for the first one, all flying birds and insects have wings. Birds and insects flap their wings as they move through the air. Therefore, wings enable flight. Is that inductive or deductive reasoning? If you guessed inductive reasoning, you would be correct. We're taking a general statement of all flying birds and insects have wings and making it into something specific. Therefore, wings enable flight. Let's try the next one. Insects generally survive mild winters better than harsh ones. Therefore, insect pests will become more problematic if global temperatures increase. Is that inductive or deductive reasoning? If you guessed inductive reasoning, again, you would be correct. So, we're taking a general statement of insects usually survive mild winters better than harsh ones into something specific about how they're going to become more problematic with increased temperatures. Let's try number 3. Chromosomes, the carriers of DNA, are distributed evenly between the daughter cells during cell division. Therefore, each daughter cell will have the same chromosome set as the mother cell. Is that inductive or deductive reasoning? If you guessed deductive reasoning, you'd be correct. We're taking something that's a very specific fact about chromosomes and generalizing it to what the daughter cell will end up having. And lastly, let's try number 4. Animals as diverse as humans, insects, and wolves all exhibit social behavior. Therefore, social behavior must have an evolutionary advantage. Inductive or deductive? If you guessed deductive reasoning, again, you'd be correct. We're taking a specific statement and making it more general. Science can also be split into basic science and applied science. Basic science will answer the question, why is this important? It will teach you all of the basics that you need to know, all of the ins and outs of how something works. Applied science takes basic science and applies it to something you can use. So basic science might teach you about white blood cell counts. And applied science, you could be in a laboratory being a technician and test somebody for their complete blood count and look at their white blood cell count and determine if it's high or low, what that means for the patient. But sometimes it's just serendipity. Sometimes things happen by complete accident and they turn into great scientific achievements. For example, the discovery of penicillin. Alexander Fleming was working in his laboratory and accidentally left a petri dish out all night. In doing so, he grew a mold that ended up killing the bacteria on his petri dish. And he used that to make penicillin, the first antibiotic. So if you ever do an experiment and it comes back a hypothesis that's supported and it's a valid scientific achievement and you want to share it, the way to do that is by writing a research article. Research articles in science are peer-reviewed, meaning that people with similar backgrounds and those who are really familiar with the field will read through and make sure that everything checks out. The abstract is the first component of the scientific article, and it will have basically a summary of the whole article. It will tell the reader what the experiment was and what the results of the experiment were. The introduction will be much like the introduction to any research paper. It will introduce the reader about why the experiment was done and maybe give a little background. The materials and methods section of a scientific article will contain any materials needed in order to duplicate the experiment and any scientific methods they used, including the way they manipulated certain data sets. The results will show the actual results of the experiment. Was this hypothesis supported or unsupported? The discussion afterwards might include things on how they would change the experiment in the future, different ways in which this experiment could have been better. They might also include information if it's a controversial topic that they're experimenting on, about that there was no monetary gain from any specific companies, to show that their work was legitimate. A conclusion will also be much like any research paper that you read, to conclude to the reader, just like the introduction. And plagiarism is any use of somebody's work that they published or wrote themselves in a way that doesn't give them credit. So throughout the course of this semester, if you have any research articles that you want to pull information from, or any websites online that you use in order to get information, be sure to paraphrase what they say. Do not use their explicit wording, and always give them credit. Use appropriate citations. Every living organism also has certain properties of life that we recognize, and we'll go through each one. Order is the first one. Living things are really complex. They can consist of one or more cells. If they have one cell, they're unicellular. If they have multiple, they're multicellular. Unicellular organisms are things like bacteria and archaea. Multicellular organisms are things like animals and plants and fungi. Many living things have levels of organization. If we look at animals or humans, we can see that at the smallest, tiniest level, we are atoms, and atoms come together to make macromolecules. Macromolecules make up organelles. Organelles are tiny organs that are inside of the cell, and they help the cell to run. So organelles make up cells. Cells then are specialized to run in specific tissues. Tissues make up organs. Organs make up organ systems, and organ systems make up the organism. So definitely keep those levels of organization in mind as we go throughout the course of the semester. Another characteristic of living things is that they have a sensitivity or they respond to stimuli, either light, touch, chemicals, you name it. Some examples of this are flowers or plants that track the sun and turn to face the sun in order to absorb the most sun that it possibly can. Our skin as humans is really sensitive to touch and to temperature. Sharks, they can sense proteins from hundreds of meters away, and it's a chemical signal. Living things are also capable of reproduction. It can be either asexual or sexual reproduction. Asexual reproduction is something that bacteria do, and some other organisms do as well. One example of asexual reproduction is called budding, and it's where another organism literally buds off of the parent organism, leaving another genetically identical organism. Essentially, they are clones of each other. In sexual reproduction, two organisms come together and exchange genetic information to make a genetically diverse organism. So, genetic diversity is a great thing. It allows us to be more adaptable to the environment, and it gives us less room for mutations to occur. And mutations cause a lot of diseases like cancer. So, the more genetically diverse we are as humans or as animals, the better it is for our species. Adaptation is something that living organisms also have. You may have heard of survival of the fittest and natural selection, and that these concepts just come together to mean that an organism is able to make great adaptations to be able to survive. One great example of this is the nautilus that's pictured here. It's older than dinosaurs. It's still alive. It's been alive for the past 500 million years because it adapted to create this slime to get away from its predators. Living organisms are also capable of growth and development. So you can see here, this is my dog Remy, as a puppy and as a 2-year-old dog now. It's encoded in our genes to grow and to develop as we age. We undergo processes of mitosis and meiosis. Another characteristic of living things is the ability to have homeostasis. Homeostasis is very important in things like nutrient transport and the maintenance of our glucose levels, of the maintenance of our pH levels and our temperature. What homeostasis means is our normal steady state. So we don't want any extreme conditions. If we were talking about temperature, homeostasis would be our normal 98.7. We wouldn't want anything higher or lower than that. A negative feedback mechanism, despite how it sounds, is actually a great thing. It helps to ensure that our body never exceeds or goes below that 98.7 degrees. We'll talk more about negative feedback mechanisms later in the course. Positive feedback mechanisms aren't always the best thing. There are some examples of it helping us, but oftentimes positive feedback mechanisms are going to act as disease states in our body. Some examples of positive feedback mechanisms working well for us are things like pregnancy and blood clotting. So the way that negative feedback works is that when your body temperature starts to exceed that 98.7 or in Celsius 37 degrees, it lets your skin and your brain know that you're too warm. The temperature regulation center in your brain sends signals to your sweat glands to start sweating. And when air blows over the sweat that you produce, your body releases heat. So in that way, your body will always be able to maintain itself. If that were a positive feedback mechanism, let's give the example of childbirth. The woman's uterus is stretching as the baby is being pushed down. So when those stretch receptors send signals to the brain, it will stimulate contractions. So the body will keep contracting until the baby is out of the body. That's an example of positive feedback. If we were to use that positive feedback mechanism for the temperature example, our sweat glands would keep producing sweat, cooling our body to levels that we don't need. So that would be considered a disease state. Another characteristic of living things is that they can process energy. In plants, they do so by photosynthesis. We'll do a whole chapter on photosynthesis later on, but converting light energy into usable energy in the plant. With humans and animals, that's converting food into energy sources. Evolution is also a characteristic of living things. It's the result of genetic diversity, and that's also a result of mutations. So we're only able to adapt as much as we mutate. Mutations are not always a bad thing. In this example, these are Galapagos finches. If you've ever heard of Charles Darwin, he went to the island of Galapagos to study these finches, and he noticed when he was there that they, although are the same species in these pictures, they have different beak shapes, and he was wondering why. And he understood that these finches adapted to different food sources. The ones with the slim beaks live high in the trees, and they have to be able to squeeze their beaks into small places in order to get their food. Whereas the finches on the left had to eat really tough nuts and be able to crack shells. So they had to have a shorter, tougher beak. So another topic that we'll talk about over the course of the semester, even later on when we get into cells, is that there are two different main types of cells, and that's prokaryotes and eukaryotes. Prokaryotes are going to be things like bacteria and archaea, and eukaryotes are going to be things like protists, fungi, animals, and plants. Prokaryotes are usually unicellular organisms, eukaryotes are usually multicellular organisms. There are a lot of differences in cell type, and we'll talk about that during the cell chapter. There's also different levels to ecosystems. So when we look at things like population, we're considering all the individuals of the same species that are living together. A community is going to be all the living things that live together in one area. An ecosystem will be all of the living things living together in that area, plus the non-living things, such as maybe the rocks and the water in this case. And then the biosphere is going to be all of the living things, non-living things, and also the atmosphere that surrounds it. One of the last topics that we're going to cover for this lecture is phylogeny. So Carl Weiss in 1970s, he separated the kingdoms into domains. Our domains are bacteria, archaea, and eukarya. Bacteria and archaea are really similar. They look similar, they just have small differences in structure. Eukarya is going to include things like animal, fungi, plants, and similar organisms. What's shown here is a phylogenetic tree, and that shows a summary of evolution. There are many branches of study that fall under biology. These are things that you could go into if you have a biology degree, or get a further degree in these subspecialties. Things like molecular biology, where you'd be looking at cells, biochemistry, microbiology, forensics, neurobiology, paleontology, where you're looking for fossils, zoology, where you're studying animals, or botany, where you're studying plants. So those are just things to keep in mind.